Great, and the good news is I'm going to catch up on some time because Stefania covered a lot of the things that I was going to say, but there's some other interesting points that we'll try to make as we go through it. I'm going to skip all of this because I think it's not important. We're a spin out from the Jenner Institute, and I think it's really important that we do recognize that a lot of the chimp adeno work has been done at Okiros, in fact almost all of it, but almost all the clinical work was done up at the Jenner. That was done with the CD63s and the Chad A3s, and when as happens in the world, when GSK bought Okiros, the ability for the academic groups to work more easily with that group was more difficult than working with a small biotech. And so because of that, the Jenner Institute developed their own chimpanzee adenovirus using one that had been isolated earlier than the ones at Okiros at the University of Pennsylvania, modify it significantly enough that it was able to obtain patent protection on that. That patent is now issued in both the US and Europe, and spun off VacCTEK to continue the ChimpAd and the MVA platform that you've heard about. So there's almost nothing original in what we're doing. It really spins off of the Okiros work. A lot of the MVA development work was actually done up at the University of Oxford. In fact, a lot of the vector development and stuff. So the MVA part that you heard from Stefania was mostly an Oxford-based. The chimpanzee adenovirus was mostly Okiros-based, and VacCTEK is mostly a follow-on base. Okay, so I don't want to take credit for anybody else's work. As she said, we're using chimpanzee adenovirus followed by MVA, and I'll go through why we're doing that. We use two adenoviruses called chimpanzee adenovirus OX1 and OX2. They're both serotype E. They give responses very similar to one of the chimpanzee adenoviruses that Stefania talked about called ChimpAd 63, which is also a group E, which has been extensively evaluated in man. There's a lot published on it, although we're catching up with ChAdOx1 now as more and more trials get done, and I'll go through those. So what are the pros and cons of a prime boost approach? And what are we doing at VacCTEK? So I think it's important to say, and I'll go ahead and say it, that there are no vaccines who have been developed based on a T-cell correlate. And there are no vaccines today that are known to work via T cells. Even the zoster vaccine has not been proven to work via T cells, which would be the most obvious candidate. So we're going to be inducing lots of CD8 positive T cells. Stefania's already showed you that. But what we don't know is whether these things do anything in actual clinical trials. So the goal of VacCTEK, to really, I think, that differentiates us from some other things, is our goal is to take these things through proof of concept and see if they work and try to find out if they do work, why they work, and what is not just the magnitude, but also the functionality of these T cells that might be important for getting them to work. Given that, we are also doing our dose finding not by magnitude, but also by functionality. So it's been shown over and over again that the doses of the magnitude of T cells are not that critical. It's the functionality. Just like the magnitude of antibody is actually not that critical if you don't have the right neutralizing antibody in it. So I think we sometimes get lost in magnitude as opposed to functionality, although I'm going to show you mostly magnitude. So we do get increased response magnitude. Importantly, when you use an MVA vector compared to two either homologous prime boosts, you also increase breadth of the response, and that's been shown in a number of human studies. I'll show you a little bit of that data. You increase the longevity of the response. I'm going to show you that data, which is really quite important. You increase the functionality of the response in one paper that's been done in humans, although that's not been done in a really great way. And you can dose spare the boost. So the dose of MVA that can be used once you prime can be much less than a dose that you would give, say, to a normal setting of an individual primed by a natural infection. What are the cons? Obviously, if you're going to do this, you have to make two products, which is a real pain and a big expense. The other issue is that MVA is a very large virus, and when you manufacture chimpanzee adenovirus, you can sterilely filter it at the end. That makes it much easier. You can't sterilely filter MVA, which means the entire process has to be done in a sterile setting, which markedly complicates the manufacturability of MVA as compared to chimpanzee adenovirus. Also MVA, because it's been adapted to avian cell lines, there's limited ways that you can manufacture it. It's very simple not simple, but it's relatively straightforward to manufacture chimpanzee adenovirus. And there is the increased regulatory complexity of using a prime boost approach. And there's the increased complexity of the regimen finding. So you've got dose finding and you've got regimen finding. And all of those things, both the dose finding and the regimen finding, we believe have not been done to the sufficient level of detail that needs to happen. So I'll just give you a few examples. They're very similar to the examples that Stefania gave. Let's see if this works. No. Does this work?
It does work, it's a bit dim though, to be fair.
This one?
Yeah, if the top button.
I can't see it.
I can't see it. Oh, there it is.
Oh, there it is, barely visible. So this is a human TB study where AD35 was followed by an MVA. Neither one gave very good response, but these are actually percentages of CD8 positive T cells in a flow assay. And to get 0.75% of your T cells in a human study is actually very, very high. It's quite high, and this is a study that was published by Helen McShane's group, which actually we funded part of it out of Eris when I ran that TB organization. And what you can see is when you prime with the adenovirus and you boost with a MVA, you can get very high responses. And if you gave 2 adenoviruses and boosted, they were quite high, higher than giving a single. However, the appropriate control, which is giving the adenovirus at the same time, was not done because this may be part of the interval between the adenovirus and the MVA as opposed to the second adenovirus. In mice, it looks like giving 2 adenoviruses does not really improve your outcome when you boost with MVA. Likewise, in this TB study that was done by Tom Hankey's group using a ChIMP-AD3, you can see that in this study, what he did is he took ChIMP and boosted it with MVA. And the interesting thing is you can see quite good diversity and breadth of response that persists out to 2 years with a single ChIMP plus MVA. If you primed with 3 DNA vaccines, then gave chimpanzee adenovirus, and then give MVA, you got essentially the same response. And the response out at 2 years, if anything, might have been slightly better with ChIMP followed by MVA than giving 3 DNAs, ChIMP and MVA. There have now been at least 5 published studies comparing DNA to MVA, DNA MVA to AD-MVA, mostly done by the VRC and the HVTN network. In every single study, AD-MVA has outperformed DNA-MVA. This is another study that's been done in humans by the group of Sara Gilbert up at Oxford using just an MVA construct and a ChAd construct, where she was looking in primed individuals. If you'll notice, the last one was in naive individuals. That was TB subjects here in Oxford that had not been exposed to BCG and had not been exposed to TB as far as we know. Here you have naturally primed individuals. They have baseline responses. So if you give them MVA, you boost them right away. So the point being is MVA is a good boost, whether you start with DNA or you start with AD. However, AD tends to be a better boost. But the important point here is to look over here and see that if you give the chimpanzee alone, you get a very nice response. It comes down, it persists out here. But now if you boost with the MVA at week 8, it goes up, and you get a much longer response. And that response now has been shown in this study, a follow-on to this study, to go out to at least a year, if not 2 years. So we think that not only do you get magnitude by using this heterologous boost, but you're going to get breadth, you're going to get longevity, and you're possibly going to get functionality, which still needs to be looked at in greater detail. So there are many clinical programs ongoing using this approach, and these are ones using AD plus MVA at Oxford. There are a mixture of the chimpanzee ChAd-OX1 that I talked about, and the chimpanzee adenovectors from Okairos, now from GSK. And I just want to tell you that there is a tremendous amount of work going on here using this system. So on that first slide, I told you for the last 20 years, people have spent an incredible amount of time looking at every possible combination of viral vector, adjuvanted protein, DNA, RNA to induce CD8 positive T cells. And the group here has consistently come down on using this approach and is now being used in malaria by Adrian Hill's group, HIV by Tom Hankey's group, RSV by Andy Pollard's group, HCV, as Stefania said, by Ellie Barnes' group, TB by Helen McShane's group, influenza, chronic hepatitis B, HPV, prostate cancer, MAGE, NY, and MERS by us at VacCTEK. So I just want to go over a little bit of that. So just to give you a sense of the magnitude difference, this is data from malaria where we can actually compare, although it's not exactly the same inserts, it's done at the same place, usually in the same laboratory. This is mostly Oxford data. I will point out that this is all mostly CHADs. These are CHADs, this is CHAD 63. But in general, we induce levels of Ellis spots, which are about 3-to-1 CD8 to CD positive T cells after the chimpanzee adenovirus, and about 50-50 when you boost with MVA. There can be much variability, but think of it as being about 50-50. So the other thing the MVA does very nicely is it balances out the CD8 to CD4 ratio, which may be very important for CD8 memory. But this, if anything, this is lower than we're seeing now. As stated, we think that using invariant chain molecular adjuvants can increase that up maybe 4 to 5-fold in humans. We know now that in recent responses without invariant chains, we're averaging 5,000 to 6,000 spots in humans, whereas when you give 3 shots of electroporated DNA at 4 milligrams, you may be able on occasion to reach 1,000 spots, which is far better than was done prior to electroporation, where we rarely got over 100 to 200 spots using DNA vectors. DNA given by injection in sort of cationic lipid formulations intradermally will also get levels in the low hundreds, but we'll never get up into, we have not seen any data that it can induce CD8 positive T cells up into the 1,000 level. Other viral vectors, measles, LCMV, a variety of vectors when given by themselves will achieve levels of about 500. The CHAD alone will also achieve levels about here. So you do not get, it's not that the CHAD is really that great compared to everything other viral vector. However, it is extremely good at priming, and it is very reproducible to make. And compared to some of the other vector systems, it can carry a much larger payload, as Stefania said. So there are advantages we think of the CHAD over some of the other systems used by some of the other biotech companies that are using viral vectors. RNA to date, there are no published data that RNA has ever induced a ex vivo CD8 positive T cell response in human, despite lots of claims. But if anybody can find that paper, please show it to me. So this is what we're doing at VacCTEK. We're trying to show this works. We're doing an influenza trial in the elderly. We're planning on an influenza challenge study in Belgium this year where we're going to use MVA alone. We will find out that insert of NP plus M1 does not induce any antibody to any of the surface proteins of flu. It will be a pure hypothesis-driven trial of whether inducing T cells to flu actually makes a difference. A hypothesis that's been around for 10 years, that's believed by many people, disbelieved by many other people. And so what we need to do is do the clinical trial and find out. That's our plan. We have a prostate cancer therapeutic that I'm going to show you some data on that's moved into phase 2 in combination with a checkpoint inhibitor using the ChIP-AD plus MVA. There's a MERS prophylactic, which I won't mention. And then we have an HPV therapeutics that we are finishing making the GMP production for now. That will move into clinical trials in the fall or late fall of this year. We are very hopeful for that product. HPV is the one place where it appears that T cells may have played a role. There's a phase 2 study that was done from Inovio and an also study done from ISA where they induced levels of CD8 positive T cells. Well, Inovio induced very low levels, ISA somewhat higher levels using long synthetic peptides. And in both of those studies, there was some clearance of CIN2-3, which is a precancerous condition, compared to placebo, but in neither case was it that appreciable. It was about 20 to 30% over placebo. We think we can do much better than that with this platform, and we're also looking to cover a lot more genotypes than were covered by those 2 vaccines. We're also moving an HBV therapeutic, this should be out here, into the clinic this fall, which will also include a chimpanzee adenovirus plus a MVA construct targeting the Asian population. And in that study, we will use low-dose checkpoint inhibitors immediately. The reason for that is that in the studies of HCV that Stefania talked about with Ellie Barnes, The most interesting thing in that study was not that they induced high levels of T cells with ChAd and MVA. The most interesting thing is that when you when you actually identified the peptides that the T cells were responding to by peptide mapping analysis, those peptides were exactly peptides that were not located in the virus that was circulating in the patient. In other words, the only T cells that were seen in those chronic infections were neoantigens in the chronic hepatitis setting. So we believe in the chronic hepatitis setting, without doing immunomodulation, a therapeutic vaccine is extremely unlikely to be efficacious. And finally, cancer. So we have gone into the group of Adrian Hill up at Oxford, prior to VacCTEK forming, had started working with the ChAd plus MVA platform. The cancer groups have finally discovered that immunology is actually quite important. As you might know, there's hundreds of billions of dollars, not tens of billions of dollars, flowing into the whole immuno-oncology sphere. And surprisingly, there's not much the cancer group does not know much about vaccines, and they didn't realize that we had a very potent platform inducing CD8s. That's now being discovered. There are many companies using chimpanzee adenovirus now in their cancer platforms, including Pfizer, GSK, Gritstone, and Johnson & Johnson's using a low seroprevalence human adenovirus. So there are multiple people that have now gained an interest in using this platform to induce CD8 positive T cells in cancer. This is a prostate-specific antigen, and if you give the chimpanzee adenovirus and the MVA, and you combine it with a PD-1 inhibitor, not a PD-L1 inhibitor, we can see very nice protection in a tumor challenge model in a paper published by the Hill group. In addition, when we used an antigen called 5T4 in a similar model, we were actually to show that when you combine the chimpanzee adenovirus plus the MVA in a 5T4 regimen and combine that with a checkpoint inhibitor, you could see marked improvement in a mouse model. I'm not going to skip that one. Given that data, what happened is that we took the Hill group through an FP7 grant, was able to manufacture the chimpanzee adenovirus 5T4. The MVA 5T4 was available from Oxford Biomedica. It was an old product called Trovax. They moved that into the clinic, and there have only been 2 papers that I'm aware of where people have been able to induce ex vivo Ellis spots to tumor antigens, and both of those come from the ISA group in Belgium. Other people have not been able to induce ex vivo Ellis spots to self-antigens. So as you'll notice here in this humans phase 1 study, this is without a checkpoint inhibitor, we were able to induce CD8 positive T cells, well, T cells, of which about a third to a half were CD8 positive when we did flow cytometry. It's a little complicated because some of those were used by expansion in vitro. But there was a 60% response rate ex vivo to a self-antigen with a single shot of chimp adeno and a single shot of MVA. This was presented last year at ASCO in the United States. So this is really unprecedented, that you can take a self-antigen and that you can give 2 shots, 1 chimp ad, 1 MVA, and you can induce relatively meaningful levels of T cells. As you'll notice to the self-antigen, we're not talking thousands, we're now talking hundreds. Okay, so whether this will be sufficient to have an effect, and whether these T cells, again, will be sufficiently functional to have an effect, needs to be studied in a clinical outcome trial. That trial has now started. The first patients have been enrolled in metastatic prostate cancer using this vaccine regimen along with a checkpoint inhibitor. So, what do I think the advantages are of a chimpanzee adenovirus prime boost? If you look at the literature, and this is actually a mixture of T cells and CD8s, but there have been, basically BioNTech was able to induce a small amount of CD4 positive T cells in their Nature paper. However, they induced no ex vivo CD8 positive T cells. If you go to that paper, they say they induced CD8 positive T cells, but that was done by doing a 15-day assay using IL-7 and IL-15 growing out the CD8 positive T cells. Neon was able to induce no CD8 positive T cells using long synthetic peptides with their neoantigens. All of the T cells induced were CD4 positive. Provenge, this is the PAP Dendritic cell has induced T cells that are on the level of 30 to 40 if you go back through those papers. And PROSVAC, which was the MVA alone, induced T cells that were in the 30 to 40 range in the minority of subjects. So we do think that this is a very promising approach for using in cancer therapy. So, why do I think this is the best approach? I've been doing T cell vaccines since 1980 something, a long time ago. That's what happens when you get old. We think we have great immunogenicity, the best in class right now. I think there could be other methodologies that will induce best in class, but today, standing here, I think I can say we have best in class. Might not be able to say that tomorrow, but I can say it today. We have almost no safety and reactogenicity. This is very different from, say, adjuvanted proteins. The ISA data, where they induce long T cell responses to long synthetic peptides, was done with essentially montanide, which is very comparable to incomplete Freund's adjuvant, which is extraordinarily reactogenic. And so I'm not sure that really is going to go too far. Manufacturing issues are pretty minor. We know that we can make these. The issues is that MVA is complicated to make because of its non-filterability at the end of production runs. So it's not fun to make MVA, but it's doable. And the cost of goods is actually quite achievable. And all of the other systems, we believe, have some shortcomings in comparison to this prime boost approach. So that was a rapid goal through what we're doing at VacCTEK. Luckily, Stefania covered, I think, a lot of the science that underlay this. And we would like to thank our partners. We're funded by some very nice investors. And we have a large number of partners we work with, including, very importantly, BARDA, who's helping to fund our flu study, manufacturing at Batavia, Emergent, and also at Advent, the former Okairos manufacturing facility in Rome. And we made our own proprietary TET-repressed cell line with Oxford Genetics. So thanks very much, and I'll take questions.